thank everybody for coming. I know it's a 5 o'clock time, so we appreciate your guys' attendance. And please, there's snacks and drinks outside. So my name's Shannon Aker from Cytiva. I'll be hosting the panel. We have great panelists up here for the topic, and the title of the topic is Added Up: 3 Things to Consider for Capacity Expansion. So we have not talked a lot internally because we want it to be fresh in some of the comments and things we say. But again, very powerful panel, a lot of years of experience in cell and gene therapy and cell and gene manufacturing, as well as just building programs, right? And so I think we're going to get a lot of intel for them when it comes down to things to consider from a capacity expansion point of view, things that, you know, from their own life and their current or previous roles where that's worked out. So instead of me doing bios, I'm going to start this way and kind of introduce everyone and have them give you a little bit of taste of their current role, their previous work, and then we'll dive into the topic. So first, Mike Paglia. So sorry, Mike, I'm going to look up your title real quick because you're just you're the boss. But I will say, so COO of Elevate Base Camp in Massachusetts. So.
Mike Paglia, technical, I worked through the technical issues. I'm the, as Shannon said, Chief Operating Officer at Elevate Bio Base Camp. It's a GMP process development technology company out of Waltham, Massachusetts. About 20 years in biotech. Started off in cell and gene therapy, where I was head of technical operations at Bluebird Bio for about 5 years, where I was responsible for all drug product, process development, and manufacturing. Did some oncolytic herpes virus production and manufacturing at a company called Oncorus before. I'll build the team and commission the facility with a great bunch of folks at Elevate Bio.
Welcome, everyone. Thank you very much for joining us. My name is Carol Houts. I'm the VP of Quality for Germ-Free Laboratories. We manufacture both modular and mobile clean room facilities, particularly for the cell and gene therapy space. My background is deep in regulatory and quality and product development, so try to bring all of that to the equation and how we serve our customers. Appreciate everyone being here.
Good afternoon. My name is Ray Stapleton. I'm the Executive Vice President for Genosha Biosciences. My background is in process development, manufacturing, and quality, with a broad spectrum of pharmaceutical products, not just the cell gene therapy. Last 6 or 7 years, I've had a pretty exclusive focus in the life sciences and biotech space.
Good afternoon. My name is Erwin Kamart. I'm an Executive Director in Process Development for Advanced. Advanced is on the verge of bringing Untitled product to commercialization. And my role is in process development. I have actually 24 years experience in that field. I recently was involved with Kite Gilead, helping the Yescarta product to also commercialization and expansion. So I have also about 10 years ultimately cell therapy experience, and for that I worked in monoclonal antibody space. Yeah, it's really great to see, I think, that we are still in the early stages of bringing cell and gene therapy to the market, and it's just great to see that we are also making progress, and really thank you for joining.
Hi everybody, Matt Hewitt, Executive Director for Scientific Solutions and Cell and Gene at Charles River Laboratories. My responsibilities are to, one, I run one of our CGT CDMO sites just outside of Baltimore, where we do a lot of process analytical development. And then one of my other responsibilities is that in the past several years, Charles River has done a number of acquisitions in the cell and gene space. So now we're arguably, I guess, have one of the better portfolios in the space in terms of end-to-end development, all the way from early discovery, antibodies, through commercial GMP manufacturing for plasmid, vector, and cell. And yeah, so happy to be here. And my expertise in general is around program development, discovery, and certainly quality, regulatory, and PD and AD. By training, I'm an immunologist.
Thanks, Matt. So I'm going to actually start with you because the panel actually has a lot of experience in historical bioprocess applications, which again have been able to jumpstart a little bit of those of us that came from the academic side and brought an academic process to a manufacturing stream and made people cry along the way. So when you look at capacity, expansion, whether that's from a startup lab or startup biotech company, or maybe somebody that's established, that's smaller and looking for large scale manufacturing. Matt, starting with you, what's one thing that you would say really focus on that's number one on your mind?
Yeah, I mean, we deal with programs that are in all different phases when they come in. Ones that are more or less race ready and others that are written on the back of a bar napkin. So, I think that in general, what I would tell folks on things to focus on, I would say maybe not focus on, I would say not neglect. I would say don't neglect your analytics. We've seen, if you look at some of the recent Nature journal papers, that look at the field in general, McKinsey just put out a nice one several weeks ago. One of the things that typically comes up when you compare cell and gene versus traditional bioprocessing and biologics is that there are many more CMC concerns from the regulators. A lot of times that centers around potency. When you're dealing with various products, some are easier to develop potency assays, and potency is regulatory speak right now for functionality. So a lot of people will somewhat bypass that, especially if they're coming from an academic institution, because during phase 1, since efficacy is not a primary endpoint, you're not required to have a potency assay. But certainly once you move into phase 2 and that becomes a primary endpoint, it's required. And if it's not at a state where it can be qualified, that can delay a filing if you end up being successful.
Anybody want to add on the analytics piece? My question would be, how much of that do you keep in-house? How much of that do you take external?
Yeah, we keep it all in-house. Certainly we work hand in hand with the folks that are holding the INDs, make sure that it's suitable and we'll be able to withstand the regulatory scrutiny that's likely to come with a program that's successful. We take an approach at Charles River where we like to keep everything in-source so that if you come to us for services, you only have to deal with one of us. You don't have to have several different external relationships manage, and sometimes things happen. If you then have to realign those stars with a bunch of external relationships, that can be quite difficult.
Mike, I'm going to come to you next because what would be your one thing, but then build on the analytics when you're looking from a CDMO point of view or from combining both a cell therapy manufacturing process with a viral vector or whatever the gene editing technology would be, and how you would handle that, or how, what advice you would give.
Yeah, I think it's the flexibility also. Adding to the analytics, it also starts with the process. So when clients come to work with Elevate Bio, we want to make sure that there's flexibility to understand what the design intent is of the process and the product. We don't take everything and just try to put it into a platform approach. We really have a thoughtful approach, and we make sure that the clients understand there's a lot of development that happens. It starts with raw materials, it will go to analytics, making sure all that's robust, but really starting with the process design, understanding what kind of product you want, the life cycle of that product, the phase of development, is it a rapid phase 3, is it an early phase clinical trial, and then making sure that platform, or that process rather, is scalable so we can meet those goals very, very rapidly. So, that integrated approach that we have at Elevate is something that making sure you understand where that state of the process is so you can design something that's effective for the client and for the product, because we know everyone gets the accelerated approvals nowadays. And making sure that you design that early so you don't have any regulatory hurdles either. So looking at the supply chain is very important early on as you design those processes.
I'm going to come back to some of these topics, but we're hitting on a lot of the major pain points. So Carol, what would be your one piece of advice for capacity expansion?
Definitely looking at the future state. It's always hard to look into the crystal ball. We have some amazing technology, some of it you've seen on the show floor, that, trying to anticipate what that's going to look like in the future, really kind of figuring out what your capacity is. And obviously that has to really align with your preclinical preclinical, your clinical program, and then obviously commercialization. And you touched on it, is that window for commercialization is so much shorter now with these therapies. So how do you approach that when you're doing facility design? You're looking at the future of how you're going to commercialize something. You're going to apply those GMPs early on. So, in my role, we're really looking at, what does a facility design have to be for a commercial GMP product? Whether you've come to us from a early stage clinical perspective, we're going to hold it to that kind of standard, because we know you're engaged with FDA all the time, trying to think about how you're going to move your program to that next step. And anything that can be done from a facility design perspective to make sure that good engineering controls are used wherever possible, it just takes that burden off already very complex processes. So,
do you feel like everyone is truly evaluating their timeline and their program, or is that a gap that, lessons learned? A lot of people don't really share that information, right? So how do you handle?
Yeah, unfortunately, so many companies are strapped with when that next round of financing is happening, that next round of funding, don't always get a chance to really look at that overall vision for, hey, we know we're getting out of the clinic, what's the next step? So ultimately, and then looking at what that capital spend is going to be, so a lot of times companies are making that choice. Do I take on a CapEx project where I'm going to put money into facility infrastructure, or can I use OpEx money to be able to then enroll more patients? So these are difficult choices that companies are making every day when it comes to trying to decide how much to invest in capacity. And sometimes taking those dollars and utilizing technology and expertise from our CDMO partners is a big option. So there's that. It gets back to some of that build versus buy discussion as well.
Great, thank you. Ray, what would be your one piece of advice?
I think that you've already heard some really nice advice from these folks. I'm going to build on a little bit what Mike was saying in scalability. And the one thing I would say from a biotech perspective, you are owning your end-to-end process. And if you focus on one, you can very easily miss the other one. So you do have to manage the complexity from patient samples in, how are you going to move material around, how are you going to move the drug product around, how are you going to get to analytical test sites, things like that. And as you scale, it's not just, are you scaling the process, it's everything around the process. It's all of your suppliers, it's all of your vendors. If you have specialty shipments, you need to make sure whoever's providing that is ready to go with you. There are cases where you may be using off-the-shelf products, but you're at a break point for one of those suppliers. So they need a heads up to increase their capacity as well, and that's going to take a little bit of time. If you have anything in your process that is unique to you, that is one to definitely focus on. Make sure that item, whatever it is, specialty reagent, is ready to go and ready to scale when you scale. Those tend to come back and bite you, but it's really making sure that you look throughout your entire process and are ready to take it all up and not just get focused on, for example, a cell expansion step or something like that.
Great. Erwin, what would be yours?
Yeah, so I will answer that also more from a process development perspective, but I definitely see what's happening in the field is also especially with autologous, because then you need to really scale out. And I would like to approach the question also from people have often going to commercialize it, so they have that first commercial facility, so to speak, ready, and they have a certain phased approach in that facility, right? So not all clean rooms are necessarily completely equipped. But I like to address the question, like if you think about globalization and you go to a second or a third facility, what's happening is that a lot of these products are, as we already heard earlier today, there's often gaps in existing unit operations. There are cost driving, cost reduction initiatives. So there's definitely people who think out what that new process is going to be. So my point is really, if you design significant process changes and where you also are developing new technology, make sure that if you plan that second facility, right, that all those timelines of the technology readiness, the process readiness, and your facility readiness, and perhaps even some improvements in business systems, all those timelines are going to come together. And that is a huge challenge. And often it is the technology development actually who may be on the rate limiting step. So keep that in mind.
Speak more to the technology development, specifically to your process development history. What advice there? You'd mentioned not be tied into, one piece of your workflow that is, it may be unique to you, or it may be unique to a specific vendor. How do you handle that, and at the same time globalization and keeping up with that from a process development standpoint?
Yeah, there's always a complex question, which really depends on really what that process is. And there are definitely some existing processes which can almost happen on one piece of equipment. They're going to be a little bit easier. There are processes which are much more complex. They are more lengthy, where you really really work with several devices, and you go from unit operation to unit operation. I think initially the technology development, there's a certain, and with technology, that's a very broad word, that could be actually a certain reagent who enables a certain step and actually enables your product. But that reagent may be highly costly, right? Or it is a certain new type of bioreactor who potentially have some advantage, but it has a monopolistic position, and the cost is going through the roof. So usually when you go to a next generation process, you want to, those high cost factors, you want to iron that out, right? So you have to come up with some cheaper alternatives. But again, that's going to drive process changes. And then the question is, you typically do that then post-BLA. So how do you know the impact on the process? So that gets rather quickly, obviously, complex. And I think it ultimately requires a really good strategy. But there's a huge challenge there. I think what we're also doing at Advanced, and what I did with my previous employers, is really make sure, and I think it's very key, that PD and a good MSET organization, and I have all that experience, so I will speak for those departments, but I think solid PD organizations is really important because they are in charge of process characterization, so they are in charge of understanding your process and your product. So they are in charge, if you make changes, either cost reductions or whatever improvements you want to make, they need to, again, generate supporting data for that. So it's really important, I think, to not underestimate the importance of those departments. And I think the most successful companies, and also recently in the CAR-T, I think you can definitely say that they had a really good PD department in there and did a really solid process characterization.
Ray, specific to you, from the Genosha standpoint, how you view some of that scalability and lessons learned, and towards maybe, and again, I don't know if globalization's in the roadmap, but standardizing some of that scalability. How do you view that and all those tied into each other, analytics, process development, and how they kind of feed into your expansion in your eyes?
It's something we spend a lot of time on. We're a much earlier stage than what Advanced is at Genosha, so we're focused on getting to that point. And to get to that point, you have to generate the data that allows you to progress through the clinic and get into some of these intensive conversations on the how and the when. I would say right now we spend a lot of time planning. One of in the first round, you heard people talk about don't put it off. Don't put it off. You have to create what I've always called that line of sight, whether it's a line of sight to pivotal, line of sight to your commercial manufacturing that operates as your true north. And then you pull into that line what Erwin is talking about. What are your major process improvements? What's the right time for that? What can you get in pre-BLA? What has to come in post-BLA? And I think, one of the things that maybe we haven't touched on a whole lot is just as important as your PD and all of the good work that Erwin and Matt were talking about, your regulatory strategy is critical. It's you take advantage of the stage you're in early on. You have a lot of ability to change. The later on, it's much more difficult to change, and you have to lay out. Mike alluded to people want to go fast. Everybody's on a fast track. Everybody wants to get to market as fast as possible. That truncates a lot of what Erwin was just talking about. Because if you skip a phase 2 and go straight to pivotal, you now lose 3 years of that critical process and analytical development, but you still need that done. And so this is what we spent a lot of time on, trying to make up for lost time, trying to be really efficient with what we're doing, being very purposeful with that process development. We can't afford to, my words, wander through intellectual curiosity. We can't go check on something because it's cool. It has to be on that true north that we have as the line of sight to get to the market.
There was a lot. Anything to add? We're all sitting here nodding, right? And so I keep thinking you guys have that history from that aspect. Which of those, analytics, process development, all the things we've talked about, which would you least likely invest from a capacity standpoint to build internally, or to look for an external partner to do this?
Owning all of it is important, but some of the analytics, but it's difficult because there's also limitations on capacity for QC and analytics out there today too. So all of those elements, I think you have the right to build and expand. It comes down to the talent, right? Mm-hmm. So understanding, getting the key talent that has not necessarily always been in cell and gene therapy, but understands the path to having well-qualified, well-validated assays. Things also, even the infrastructure, integrating all of that. I know we'll probably talk about it and touch on systems as well. Making a decision early on the systems that you integrate very early on an expansion project, so you can take those analytics, you can take that data, and actually make sense of it later. For example, at Elevate, we very early on, before the plant was even built, we selected our ERP because we knew we were going to be in cell and gene therapy, autologous cell therapy. Clients were going to have multiple patients in a room at the same time, a lot of throughput, and purpose-built systems to be able to make sure you could support that, rather than repurposing something that does not necessarily work, but it's what everybody else uses. So I think trying to think about what you would outsource. There are several components. There's a lot of great companies doing everything. Smaller companies can't do everything. That's why there's these new models like what we're doing to be able to offer that, even if it's bits and pieces. And some companies in my experience, they like to own some of that process development always, because it always ties to the intellectual property and the know-how, and they oftentimes like to own some of that, but we try to encourage them that there's ways to work with us to be able to actually accelerate, and yours is yours.
Mm-hmm. Matt, back to the analytics piece and how all that wraps in. Any words of wisdom there on how somebody would view it? Maybe you could view it from somebody that is established versus somebody that's a startup. That would probably be helpful.
Yeah, I think in general in the field we see a lot of, if you look at some of the surveys out there, like the CRB Horizon survey as an example of what people are generally looking for when they come to us, and to get a bit broader than analytics, it's generally around general process analytical development and optimization. That's where the biggest gap is, and I don't think that's necessarily around developing a potency assay. It's more about how do you develop a potency assay that is suitable for GMP and commercial. So I would say, I think that we just need to seek out that help, and obviously that's why we're here. That's why a lot of us have been doing this for longer than some of us want to admit. And I think, in the capacity around that, again, more unified, simpler solutions. So, co-locating testing at your CDMO site so you can affect, near real-time product release, so you're not waiting on a conditional release assay. Logistics right now is not ideal. So, you can imagine, and I've been in those shoes before, where I've been up all night on the phone with the lab because we have to dose a patient by 11 a.m. and we're waiting for the results of a conditional release assay. And it's also in a locale that is, 7 hours ahead of us. So, you're pulling all-nighters, and that's not fun. So I think trying to simplify and, somewhat centralize that QC testing at the site of manufacture makes sense. It brings up a lot of other questions, though, about how you do that efficiently at scale and, across different geographies. And that links into everything everybody else has been talking about as we scale out. And I think it's probably important to remember that of a lot of the liquid or heme tumors that we're dealing with represent around 6% of tumors year over year. So we haven't even really scratched the surface yet. So we don't even really know what this is going to look like. And I think that's another thing is, I say, and it's the reason why I like a lot of the solutions from Carol and Germ Free is, I've been doing this for a while. I don't know what the manufacturing is going to look like in 5 years from now. And if we're all just doing stick builds left and right, we might be in a world of hurt in 5 years when everything needs to change. Maybe just one other thing is, I think it's laudable everybody wants to maintain their independence given the imbalance between demand and supply for, plasmid, vector, and cell therapy manufacturing facilities. But, if we all start building our own facilities, we are going to make a talent problem much worse.
Right.
And I think what you're going to end up with as well is that the CapEx costs are the cheap part. Your OpEx is what's really going to eat you. And so I would just think about that if you decide to go down that building a manufacturing facility route, understanding what your actual burn is, a year from now and add that up. I can almost guarantee you it's going to be far less than coming to Elevate or, Charles River or some of the other CDMOs that are out there.
Mm-hmm.
Scalability keeps coming up. I've got a list of questions I'm picking. We've hit on so many topics we could talk another hour about, right?
Yeah, so can we talk just a little bit about, Matt made a couple really important points, and being a smaller biotech, I think I can probably build on some of his points pretty well. One of the things I spend a lot of time within our organization doing is talk about reality. Make sure you understand what your internal capabilities are and be honest with yourself. Whether it's infrastructure for a PD lab or analytical lab, whether it's your HR capabilities, be honest. If you're a small group, you have one program, you have two programs, it's fairly unlikely you're going to have a deep bench in a lot of these spaces. Wherein you can reach out to Matt or some of these providers, Mike, and draw on their experience to bolster what you're doing. And that's going to make you go much faster, and that will help address that burn rate, right? Because you're going to get more done in a shorter amount of time. One of the things I see trip more people up is we can do it, right? And just be really honest with yourself if you can or cannot do it. There are good people out there that can help, and there's a reason that this area is growing. But, Matt alluded to a talent drain. We have, I think I saw something this last year, there's something like 500 cell and gene therapy companies trying to run clinical trials right now in the world. They're just not staffing for 500 cell and gene therapies to have experts, right? When you have a concentrated use point in some of these CDMOs, especially early on, they can really help you jumpstart where you're going rather than you're trying to do it on your own. I think that's pretty important to understand where you are and be honest with yourself about it.
Staffing. How much does that factor into where you choose and which areas you choose to do the capacity expansion in and which you don't? Obviously, there's a staffing crunch everywhere for our field. Any advice?
I mean, I might, so in a prior life, and a couple of my former colleagues are in the audience, I was with Lonza, and I did a lot of work with the cocoon technology, and helped launch that with a couple of the folks here. And I think that there was a really good point made earlier around flexibility and process. I'm always kind of conflicted about when you decide to introduce automation, because if you do it too early, you can really box yourself in. And we see this in a lot of again, when we go out and ask people, when do you implement it? A lot of folks are saying after phase 1, which I generally agree with, because during phase 1 you're doing a lot of dose escalations, and you don't really know what cells numbers you need. And where I'm going with this is that I think eventually you do need automation, because then the requirements of the folks that are now doing manufacturing is hopefully simplified. Mm-hmm. And so just like we're trying to increase patient access to these therapies through COGS reductions, we also need to keep in mind that given the talent crunch, we need to increase the accessibility for people to actually be able to do these processes for us in a GMP environment. Moving to a closed automated system is great, because now, once you're qualified as closed, you can move into a lower grade clean room space, which means that your gowning requirements change, people are much more comfortable. You move to something around CLIA, right? CLIA certification, where you need a high school degree to do it. I think that's where we have to head, and I think that will help a lot with the talent crunch we're seeing, because we need a lot more of the people executing manufacturing. I mean, there's plenty of thinkers up here, but I think we do need some people that are, executing on the processes, as well as when you decide to scale, activating sites means that you transfer a software package with a piece of equipment, rather than sending one of your main process development people or MSAT tech services to each site to train them on a specific step that has some nuance to it that you can't explain through an SOP.
I don't think the region is necessarily the most important thing either. I mean, you can always find the talented people to do the work, and the automation obviously ultimately is where you want to go. But it's the retention also. You get them there, you get them trained, especially on the manufacturing end or the analytics, and then they're moving on to somewhere else in the company, hopefully, right? You want to retain your great folks, or there's a lot of competition. So sometimes it's better actually to not necessarily go to one of the hubs because there's so many opportunity for folks to move around. Yeah, I think the retention is also the next best, because everyone experiences it, is making sure your talented staff, keep them happy and they stay around and work on these novel therapies for a long time.
Back to the scalability, which factors into the labor. How, again, from the expansion point of view, when you put in automation, how much do you factor in, I use the air quote, all-in-one solutions that may not be flexible versus scalable solutions? How do you guys view that within your processes as you move through phase 1, 2, 3? Obviously some of them are, have limitations within digitization, getting electronic information, real-time information out of those, versus some that have that capability. How soon do you evaluate that within the process, within your processes, or how soon would you give advice to somebody to look at those? Because again, as you mentioned, Erwin, you will get tied in at some point once you get to phase 2, where you can't go back and make some of those larger changes that may affect your scalability.
Yeah, I think ultimately that you do that early on, and you do that actually more frequently, right? So you can make a plan, and you look at kind of all the factors which you think can reduce cost, or which are important to select certain equipment or new technology. But I think as you're going, the technology that goes through milestones, right? So let's say you get it to proof of concept, and then you're still going to revisit that, because what happens is that your first process, so to speak, that brought you to phase 1, that also can actually evolve over time. And so if you're developing a little bit more transformative, basically, you're going to actually compete with your own baseline process, which incremental improves. So I think rather frequently kind of revisiting, do we still have all the drivers in place? Are there still cost reductions? Are we still talking about reduction of manual touch points? Is actually labor reduced? Every milestone, I think you have to almost revisit that. Yeah, and just to make sure you're still on track.
You mentioned reducing touch points. How much would the five of you put focus on as we're moving into the meta world, right? We're all going to have a meta version of ourselves, and things become more electronic, ESOPs, electronic batch records. There's been a low adoption rate over time for those technologies. How important would you suggest those be for somebody that's entering the field? And then also, what would you want to see to push those into where we all know that they probably will end up being? But again, how do you view that from a low adoption rate? Open to anyone.
I mean, it's come a long way even the last 2 years. I mean, there's some really great solutions out there where you can inexpensively get paper on glass. You know, something that's digitized, can be validated, it works well. And then there's definitely a huge benefit of a full-blown EBR. When you have a process that's defined, you know, we have both, and it's interesting because the implementation is costly. You don't necessarily go full-blown EBR until the process supports it and you're at a later phase clinical development, because it is very costly. But once it's implemented, it starts to reduce the risk, especially with new operators in the rooms, new operators in the suites. It takes a lot of the question out of it. You still train them. It's very important that they're educated and they're trained well, but it takes out a lot of the variability when it's a full EBR at some point. But there's some really great low-cost solutions now to dip your toe in the water.
I think you said something like 3 or 4 times that was really critical, and that is the lower cost solutions. You know, back in the day, I remember when we were implementing LIMS systems, you know, it was quite some time ago, and I think we're finally at that place with EBR now, but it's the low cost and the complexity around implementation that I think has really been a barrier. But it really is the only way that you're going to control unit operations to the level that you need to be able to maybe minimize some of that staffing burden, and the way you'll get enough repeatability and to be able to scale. So I'm excited at this time because of where we are with EBR, knowing how successful LIMS has been over the years. I don't know where we would be without that now. It's sort of, you know, a legacy of the past, but certainly we're benefiting from it.
I think there's 2 kind of running themes coming out of this discussion other than scale up, right? That's kind of guaranteed. One of them is timing, and the other one is process knowledge. You're on a continuum as you constantly build your process knowledge, which drives the timing that makes things correct, like Mike was alluding to. You can't go full EBR when you haven't fully defined your process and you're not exactly sure what you're going to do. So they're great technologies, all in good time. That's kind of how I look at them. And, you know, some of this space is a maturation of the business relationship with the agency. A lot of early adopters in the electronic record space maybe didn't have the greatest success. And there were some obstacles to implementation. And I think, as Mike alluded to, as the technology has matured and evolved, the agency's matured and evolved, and we as a therapeutic area has matured and evolved, it's starting to feel like a good time. So now you start to implement it on your timeline and your process when you get the most bang for the buck. Again, go back to what we were talking about earlier. When you create that line of sight and you're going through early clinical to late clinical to commercial, all of these things will have really important milestones on that line.
Yeah, I think also from going back to that question about labor and the manual touch points, I think it is, I see a lot of processes still will have the unit operation. So then I think per unit operation you can ask yourself, like, yeah, where are the manual touch points? I think if you can shift them to, let's say, the beginning, that you set up equipment and then the equipment actually doing its job and automate it, and at the end you take it back on and you go to the next. It's really important. So, but the intermediate touch point, if a machine after an hour doing something asks you to open a clamp, yeah, that's super annoying, right? So that, because then the operator needs to stay with that unit operation. So there's something to be mindful. And then I think the manual touch points is also a little bit about what was said earlier about, I think with the operator training, is if that operator is doing something extremely critical, which you can only train by operator to operator training, yeah, that's also something you want to take out, right, as much as possible.
Yeah, I mean, I think it's, I personally think the pace of the technology has been good. I think, other than stem cell transplants, there's really nothing else prior to cell and gene that was truly personalized. So when you're doing a 20,000 liter reactor or you're formulating small molecules, I mean, paper batch record might do, because how often do you do it? But when we're talking about running 10, 12 processes a week, incorporating, 4 teams of, anywhere between 3 and 20 people apiece, now you're talking about some serious paperwork. And I mean, my writing's not that great, so, we're gonna have other people interpret everybody else's writing, 7 years from now. Seems kind of risky. So I think that we haven't really had the push until the autologous therapies, and specifically Kim Ryan, Yescarta, when they got approved and showed that they could get across the finish line. I think that put this into focus, is that we need it. And so I think in the last 4 or 5 years, we've made tremendous progress. And I think what we're seeing now is people pretty excited about getting rid of paper. And I think that's, we're definitely heading that way. I'm sure everybody else up here is heading that way in one shape, form or another. And, next 5 years, I'm sure we'll be even, 2 years ago, I thought there was no chance that regulators were going to be okay with the cloud, right? Let's just throw everything out there. And now we're seeing they're fine with it, so they are adapting.
Thank you guys very much. Maybe we can give a round of applause for the panelists.